Very happy to be invited to give this talk. Right, yes. Okay, so a slight change to my title. I've called it now Less is More. But I'm going to talk about biased agonism, which some of you may be familiar with, many of you may not be. So we've heard from Fiona and also David about the different types of GLP-1 receptor agonists that have emerged over the past 20 or so years. I've grouped them into the three types shown here. So we have the injectable types, injectable GLP-1 receptor monoagonists, which are sort of typified by semaglutide. We also have the orally available GLP-1s, again semaglutide being the example which is currently available, but a few more on the horizon, as Fiona mentioned. And really the field is now heading towards this strategy of targeting more than one receptor at the same time. But these sort of multi-hormone receptor agonists are heavily based on GLP-1 as the foundation, and then you add in extra receptors that you target at the same time to either drive additional weight loss or add additional beneficial effects that you may want. But I think because GLP-1 is really the basis for these treatments, it's still legitimate to ask, have we actually established the best way to target the GLP-1 receptor? So that's what I have sort of been, one of the things I've been working on for the past few years. So yeah, around 10 years ago when I was starting my PhD, I was testing a bunch of different GLP-1 receptor agonists that we'd developed in the lab. I was doing sort of in vitro pharmacology assays where we look at responses, and there are a number of examples, such as the red one here, which is a partial agonist, so it produces a reduced maximal response even at high concentrations. So I would have assumed that those would be pretty clinically ineffective or therapeutically ineffective, but to our surprise, or to my surprise at least, we found that those were the ones which worked better when we went in vivo. So they had higher sort of therapeutic efficacy in our in vivo pharmacology models, and potentially also a better therapeutic window as well. So by that I mean sort of signs of improved tolerability, and this is compared to the more standard full agonists, which otherwise I would have expected would be better. So why is this? So we think this could be related to something called biased agonism. As you've heard, the GLP-1 receptor is a G protein coupled receptor. When it's activated, it in turn activates G proteins, and they drive the downstream effects of GLP-1 in beta cells, in neurons, so they're coupled to insulin release, appetite suppression, etc. But there's also another part to the intracellular response, and I would highlight the beta-arrestins. So these are molecules which are sort of classically known for switching receptors off. So they sit there under the receptor, prevent the G protein from binding, and they desensitize the receptor, and they drive it into endosomes, and then ultimately it's trafficked to lysosomes where it will be destroyed. So this is sort of like a negative regulator of receptor function. And what we typically found was that where you have a ligand with sort of reduced G protein coupling, shown there, that's almost always accompanied by concurrent sort of even more dramatic reductions in how well it recruits the beta-arrestins. So hopefully you can see the graph on the right there. So even though these partial agonists, the red ones, show reduced G protein coupling, they're actually biased in favour of the G proteins because they have so little beta-arrestin recruitment. And because we think of these beta-arrestins as negative regulators of receptor function, then this might allow a sort of weak agonist to sort of escape its restraints and actually end up working better. And certainly that's what we found in our in vivo work. Just a little bit more about what these ligands are. So as you've heard, the vast majority of the GLP-1 receptor agonists that are available are peptides. They're based on either the native GLP-1 peptide hormone sequence or in some cases, exendin-4, which was the original GLP-1 receptor agonist that was approved. So we've done some work using exendin-4 analogues as our starting point, and we found that actually just making really very small changes to the N-terminus of the receptor, which is the part sort of at the top left there, you could quite profoundly affect the signalling profile that you get with these ligands, so as shown here. And that actually makes sense because, I don't know if I can quite point to it, but so basically that's this part of the molecule down here, shown there, and that makes quite sort of exquisitely sensitive interactions with the receptor itself to drive activation. So even just very small changes there can lead to quite large changes in how the ligand signals, and in this case leads to development of biased signaling. Just as a sort of a visual representation of how these ligands can behave differently, in this case in pancreatic islets, this was a study that was done in collaboration with David Hodson and Johannes Breukhagen. Essentially, we've added the fluorophore in the same way as David was just talking about to these biased ligands. And now we can see that the one on the left, which is a sort of typical full agonist, has a very punctate appearance because it's been internalized into the cells. Whereas on the right, we have a typical G-protein biased partial agonist, which very much remains at the cell membrane where it continues to signal over and over again. And that seems to allow it to work better for longer. So just some in vivo data. I mean, there are lots of, or many examples I could have shown which represent this phenomenon. These are some data that were published by Charlotte Hines from our group last year. So here she's compared semaglutide, our sort of reference full agonist, against a partial G-protein biased agonist with a long half-life called SRB107. And this is in diet-induced obese mice. You can clearly see that over time there's greater weight loss with the biased agonist. And interestingly to me, that sort of only really kicks in after a few days and becomes more prominent sort of later into the study. And that's typically what we see with these ligands, actually. There are also quite major improvements in blood glucose control with this ligand class. And then perhaps sort of more interestingly, this hasn't quite come across properly, but so this here is the results of a behavioural study where we basically examine the mice for signs or behaviours which are considered to be sort of equivalent to nausea in humans, because mice don't vomit, so it's difficult to make those assessments, but this is one way that we find useful. So essentially we found that the biased agonist shows a reduced tendency to drive these potential nausea-associated behaviours. So this may mean that this group of ligands is going to be more tolerable, and we can think of it as maybe increasing the therapeutic window. And there are some interesting possibilities as to why that might be, which I don't have time to go into today. So pretty much all of our work has been done using preclinical models. Is this relevant to humans? Well, currently we don't know, but I think it's worth flagging that some of the, either promising, including orforglipron, which is this orally available small molecule drug, and tirzepatide, which is the current class leader, at least in terms of the approved drugs, both show this class of GLP-1 receptor signaling, and that's distinct from semaglutide, which is more of a traditional full agonist. So there are potentially some signs that this could be beneficial, but there are too many alternative explanations for us to be confident about that. So the take-home message, I think, from the work that we and others have done is that this may be a route to improve how we therapeutically target a GLP-1 receptor, both in terms of efficacy and tolerability. At least that seems to be the case in the preclinical models. We really need more human data to be confident about this in humans. And I've just put up a few questions here which I think are relevant to this particular field of research. I've said the first one, does it work in humans or not? We don't yet know. may be sort of relevant to, you know, long-term treatment, which would be, I guess, applicable to this audience. You know, the standard GLP-1s have been around for 20 years plus, so we have a huge amount of data of their long-term effects. But these newer biased GLP-1s, we don't know yet. We're not expecting any issues, but it's always possible. They may show tissue-selective effects. That's something I haven't talked about today. But I guess, you know, the other thing is this seems to work well for GLP-1. Does it work well for other hormone receptor targets? We don't know. A number of pharma companies have started to target some of the other hormone receptors in this class as well, and some of the results look quite promising. So that's it from me. Lots of people have contributed to this work, some of whom are shown on the slide, and we'll acknowledge the funders as well. And I think that's everything. Happy to take some questions.